Hello, I hope everyone is well. My name is Van Loo, and I'm an early career investigator at the University of Western Ontario. Firstly, I'd like to thank the Gairdner Foundation for organizing this wonderful event and for recognizing the achievements of so many brilliant scientists. Also, thank you for the opportunity to present and share some of my research. It is my pleasure to follow this year's Gairdner International Award laureates, Drs. Daniel Drucker, Dr. Joel Habener, and Dr. Jens Holst. Collectively, their work has led to the discovery of the peptide hormones glucagon-like peptide 1 and 2, which are primarily produced and released from the gut. Their research, both independently and collaboratively, has elucidated the biology and physiological function of these hormones. They uncovered the involvement of GLP-1 in regulating blood glucose levels by enhancing glucose-stimulated insulin secretion, and the role of GLP-2 in stimulating proliferation and enhancing nutrient absorption in the gut. More impressively, they have translated their work to test the use of these gut hormones for the treatment of metabolic and gastrointestinal diseases such as type 2 diabetes and short bowel disease. Their work has expanded a new branch of endocrinology research centered around the contribution of gut hormones to overall human health. They continue to make significant contributions to the field, creating models and studying mechanisms that regulate gut hormone secretion. My own research has certainly benefited and has been guided by their work. Now, my research focuses on the molecular mechanisms governing GLP-1 and 2 release from specialized cells in the gut, the enteroendocrine cells. I'm particularly interested in the role of ion channels in this process. Now, to study this, I have performed electrophysiological measurements on isolated enteroendocrine cells, as illustrated in this photo of a glass patch pipette placed on a GLP-1 releasing enteroendocrine cell. Now, using these electrophysiological techniques, we are able to study enteroendocrine cells because, like neurons and muscle cells, enteroendocrine cells are electrically active. The membrane potential, or the voltage across the cell membrane of an enteroendocrine cell, increases when we inject current. And with a sufficient stimulus, they will generate these action potential spikes. Now, I have determined that the voltage-gated calcium channel, the PQ type, are responsible for generating these action potential spikes, as repeating this protocol in the presence of a selective PQ-type calcium channel blocker, agatoxin 4A, blocks the generation of these action potentials. Thus, PQ-type calcium channels are responsible for generating the electrical activity in GLP-1 and 2 releasing cells. Now, not only are enteroendocrine cells electrically excitable cells, but their excitability can be modulated by nutrients. Whilst investigating the glucose-sensing mechanisms of GLP-1 and 2 releasing cells, we found that application of high glucose triggered an increase in the membrane potential recorded and an increase in the firing of action potential spikes. Now, this graph summarizes the effect of high glucose on the firing rate of several cells and the reversibility of this effect. And as expected, GLP-1 secretion was enhanced with high glucose. So to summarize our work elucidating the mechanism of glucose-stimulated hormone release, glucose is transported into enteroendocrine cells by the sodium-glucose cotransporter SGLT1. Now, the movement of sodium across the transporter with glucose produces a small electrogenic current, which is sufficient to increase the excitability of enteroendocrine cells, and that this increased excitability is sufficient to trigger an increased release of GLP-1 and 2. Now, we were able to directly demonstrate the importance of ion channels in mediating the excitability and secretion of gut hormones, as application of the selective calcium channel blocker I mentioned previously, agatoxin 4A, was able to attenuate the enhancement of GLP-1 release by high glucose. So, the action of agatoxin on PQ-type calcium channels, which are responsible for generating action potentials, was able to regulate gut hormone release from enteroendocrine cells. In other mechanistic studies, we have found that different nutrient-sensing pathways recruit other ion channels to modulate enteroendocrine cell excitability and function. One example are the pathways responsive to long-chain fatty acids. Now we know that fatty acids can activate G protein-coupled receptors of the free fatty acid receptor family, and from expression data, there is enriched expression of the free fatty acid receptor 1, or FFAR1, in GLP-1 and 2 releasing enteroendocrine cells. Now I tested the effect of an FFA1 agonist on membrane excitability. And as you can see, application of this agonist increased the electrical activity of the cell by increasing the membrane potential of the cell, and that this depolarization triggered action potentials, which is observed as an increase in the number of spikes recorded. Now this graph shows a summary from several cells treated with the receptor agonist and the significant increase in membrane potential recorded. So to summarize, activation of FFA1 receptors depolarized enteroendocrine cells, and this depolarization triggered the cell to fire more action potentials. I was able to further elucidate the mechanism of long-chain fatty acid signaling, and I determined the ion channel responsible for this increased excitability. To do this, I've recorded the current produced and its voltage-dependent properties following FFA1 activation. Now this current was blocked by the drug PI3, which is selective for the transient receptor potential cation channel subtype C3 or TRPC3. Furthermore, addition of this selective blocker attenuated the increase in GLP-1 secretion following activation of FFA1 receptors. So to summarize, the channel opened following FFA1 activation is TRPC3, and PI3, by blocking TRPC3 channels, reduces the effect of these channels on enhancing gut hormone secretion, thus demonstrating the ability of TRPC3 channels to modulate enteroendocrine cell excitability and function. Recently, we've expanded our studies to investigate nutrient-sensing mechanisms in human GLP-1 and 2 releasing enteroendocrine cells using a novel model, the human intestinal organoids. Now in this model, GLP-1 releasing cells are labeled by CRISPR knock-in of a fluorescent reporter into the gene that encodes for GLP-1 and 2, the proglucagon gene. Now this photo on the right shows a human intestinal organoid with GLP-1 releasing enteroendocrine cells labeled with venous fluorescent protein. Utilizing this model, we were able to confirm that, similar to our rodent studies, human GLP-1 releasing enteroendocrine cells express FFAR1, and application of an FFA1 agonist, this time a new synthetic agonist, AM1638, produces an increase in membrane voltage, illustrated in this sample recording and across several cells shown in this graph. An application of this FFA1 agonist also increased GLP-1 secretion, which was dose-dependent. Finally, we've been able to apply our methodology to create new human organoid models and expand our studies to other classes of human enteroendocrine cells. Excitingly, I found that different classes of enteroendocrine cells behave differently in terms of their excitability and action potential firing pattern, meaning given the same stimulus, this class of ghrelin and modulin secreting enteroendocrine cell found in the upper small intestine tends to fire several action potentials, whereas the GLP-1 secreting enteroendocrine cell we've been studying fires just a single action potential. This suggests that different enteroendocrine cell types express a different complement of ion channels, and we will continue to study these channels and determine if we can exploit this to target one class of enteroendocrine cell over another. Thank you very much for your attention. I'd like to acknowledge the many people I've worked with on the projects I've described today in my former lab at the University of Cambridge and in my current lab here at Western. Again, my congratulations to this year's Gairdner International Award laureates, Drs. Drucker, Habener, and Holst. Thank you for being active leaders and engaging scientists in our field. All of the best.